today we are dealing with the part 4 of the neurophysiology, and today we will be discussing about the PNS system and how the nervous system responds to an injury, etc. So the supporting cells of the PNS system are called the schwaan cells, and they are helpful in formation of myelin sheath and provide insulation. So we talked about the oligodendrocytes which provide the myelin sheath in the neurons of central nervous system, but here the schwaan cells will be providing the myelin sheath. So I hope you remember this picture. So you can see the schwaan cells, and these how the cells wrap around the axons to form the myelin sheath. So let's talk more about the myelin nerve fibers. According to the insulation or the nakedness of neurons, the fibers can be divided into myelinated and unmyelinated. That is, the neurons which have myelin sheath are called myelinated, and simply those cells or those neurons which doesn't have myelin is called unmyelinated neurons. And the insulation which are provided by the sheaths may either in the form of a lipid layer, which is a thin tubular covering of the neurolemma, or simply the schwaan sheath of the cell. For knowing more about the nerve fibers, we can see this. So the axon is composed of delicate neurofibrils which are embedded in the axoplasm along with the mitochondria and endoplasmic reticulum, and the cell membrane covering that axon covers a part called axolemma, which separates the axon from the surrounding structure. So axolemma is surrounded by a thick myelin sheath made up of whom? The schwaan cells. And this structure possesses the axon to be highly refractive and provides that white color to the axon. Due to such schwaan cells, the neurons will be appearing as a white color. So coming to the structure of the myelin sheath, the myelin sheath is made up of a lipid called sphingomyelin. You have to remember that because sphingomyelin we will be discussing in throughout the course of the veterinary, like in biochemistry and all. So what it is made up of a lipid called sphingomyelin, and you know that there is knots of Ranvier, right? These knots of Ranvier are what? These are the constriction points present at the axon, like they made by the schwaan cells sheath, that is myelin sheath. And at each knot there will be the constriction. That is what it is talking about. It is the process of myelin formation. So how it is done? It is done by the process of crowding and excessive growth of the schwaan cells encasing the whole length of the axon. And the cell membrane of the schwaan cells surround the axons, which will be rotating in a manner to wrap around the axons and form closely packed helically arranged layers of double membrane. So here in this picture you can see the schwaan cells are wrapped around it helically to form the myelin sheath. So coming more, the each membrane will be composed of a bilipid layer sandwiched between the protein layer, because they are wrapping around, right? And this thickness of the myelin sheath is determined by the number of membrane layers wrapped around the axon. Simply, there is nothing to explain. And the myelination of the sensory tract precedes the motor tract. So always the myelination will be preceding the sensory tract, and it can be influenced by certain factors such as vitamin B12, folic acid, thyroxine hormones, etc. So let's discuss about the stages of myelination. So first stage begins with the enclosure of the axon with myelin forming cells, that are schwaan cells. And this mesoaxon is formed by the union of enveloping processes from the outer surface. So the enveloping processes from the outer surface union will cause or will form the mesoaxon. And the elongation of mesoaxon will succeed after this in a spiral manner. As the spiral turns around, the cytoplasm of the mesoaxon will be lost, and thus it will be getting more tight. So let's discuss about the reaction of neural tissue to the injury. I hope till now it's clear. If anybody have any doubts, let me know in the comments. So what is basically an axon degeneration? We can see that when an axon is interrupted, the distal segment will be getting degenerated, and that degenerating part or that degeneration is called as the Wallerian degeneration. It can be asked for your 1 mark or short essays, etc. And if the cell body is undamaged, the regeneration and return of function can be occurring. We all know that nervous tissue will not get regenerated, but under circumstances there is regeneration at some extent is possible. So let's see how the regenerations are occurring. So the regeneration steps will be discussed in the next video. Thank you for watching. If you like this video, don't forget to subscribe my channel, and thank you.